for just a certain amount of sodium to be moved against concentration and also against that charge difference. Okay, how do we know about this transporter? Well, there's the picture of the structure that I showed you. We know a lot about that. But we also know that we can trap this molecule in the E1 and E2 states using different inhibitors. For instance, we could use different forms of ATP. There are forms that cannot be hydrolyzed. Or there are inhibitors of ATP binding or inhibitors of dephosphorylation. But there are also inhibitors of just this state transition here. For instance, these drugs, Uabane, and the other guy here, digitalis, or also known as divoxin, I can't say that, which is from this plant, Digitalis purpurea, which is well known in any sorcerer's book as a very good toxin. It's one of the most toxic plants in nature. If you eat these flowers, you're not going to survive. So what's in there? There is this molecule and it binds here, and for Uabane we know that it actually blocks potassium binding. And potassium binding is required to ultimately get to the dephosphorylation. So if you block that, the molecule is trapped in one state, and it functions by going from E1 to E2 to E1 to E2, on and on and on, and that cycle is blocked. Okay, so these are inhibitors, and Uabane is found in the bark of some African plants, and there are tribes that put this stuff on their arrows, and they use it to actually hunt. They kill their prey. Okay, Uabane. You should know Uabane as an inhibitor of the P-class pump. Uabane. That's the movie with Robert De Niro? No, it's not. Al Pacino. It's called Scent of a Woman. And the guy always says, "Wah!" in that movie. And it always makes me think of Uabane. It has nothing to do with this drug, but I know this is one way in which we will not forget. Uabane is an inhibitor of the sodium-potassium pump. Now these inhibitors actually have medical relevance. They are used, especially digitalis has been used for that purpose. I think Uabane also. They're used to inhibit this pump as it sits in the heart. The heart, or cells, they need this sodium-potassium gradient. But if you give low concentrations, you actually inhibit this pump a little bit. And what happens then is that it becomes a little bit more easy for those people to have their contractions of their heart cells. This is because you need an inversion of the membrane potential, we'll see later, over the cell membrane. And the membrane potential is kept steep by this pump. So if you have patients who have trouble in inverting their membrane potential, getting an action potential in their heart, then it helps to slightly inhibit this pump. Of course, not too much, because then you die. So it's interesting, right? So you see that a very toxic molecule, under some conditions, can actually temporarily save lives. Okay, so here we have our major transport pathways. And if I show you this diagram, you should be able to tell me that this flat line here should make you think that you have a closed channel. Or if you have this very shallow climbing line, then you're suspecting that you have simple diffusion, not protein mediated. Or if you have this saturating curve here, this curve indicating saturation, then we have either a carrier or a pump. And if ATP is also required, then we think we have a pump. And if we have this very steep curve, not saturated, steep line, straight line, not saturated, then we think we have a channel or a pore. Favorite exam question is? I see it. I see it. Okay. How are we doing? Okay, so we have membrane transport pathways. Simple osmosis, simple diffusion osmosis is one of those cases. Then we have channels. Channels can be gated. Channels can be specific, but channels are not saturated. And we have carriers. We have uniporters and we have symporters. And the symporters, or co-transporters, they allow secondary active transport. And then we have the pumps. Another question you should really ask is, why do we care? Right? Why do we care about all these different transport systems? And I'll give you a very straight answer. We would not be here without these transporters. There wouldn't even be a single cell. And let me tell you why. So, most of transport systems we're now going to look at. I'd like you to understand how these different types of transports are used together in order to make functional cellular systems that make sense. So we'll see that we need transporters to counter what we know as the Dolan effect. I'll explain that. To control the amount of water and the amount of ions, etc. that is in cells. We need transporters in order to have actual energy metabolism. The whole process of making ATP depends on transport systems. And then, in order for us to communicate, we need transporters, channels, and pumps. Okay. So, let's look at the Dolan effect. This is the primary reason that all cells need transporters, and especially we need pumps. So let's just think about, here we have a simple cell, a membrane enclosed compartment, and there are ions, or salts, that dissociate into ions that are positively and negatively charged particles. And under equilibrium, we are not under equilibrium, but under equilibrium we would have the same concentration inside and outside. Everybody's happy. However, this is not life. Life has macromolecules. There are macromolecules within the cell. DNA, RNA, proteins. Those are charged macromolecules, and those charged macromolecules bind ions, causing the equilibrium in the cell to be non-existent. So any ion that is bound by proteins here, or by nucleic acids, is going to be replaced by an ion from the environment. So because of the presence of charged macromolecules, cells attract excess ions from the environment. So we know that this happens. Excess ions moving from the environment, attracted by the charged macromolecules that are part of life. They're in the cells. So if that's the case, then water will follow by osmosis. Water will follow by osmosis. Every cell will burst. And there's no membrane enclosed compartment anymore. We're all exploding, we're all dead. We are not, because life has found the solution to this, what we call the Dolan effect. It's a simple physical-chemical law that dictates that we should have a solution to the problem of osmosis caused by excess ions getting into our cells. So what can we do? There are three possible solutions for life. Very fundamental, every cell must have it. So, we have it just in text. What's the Dolan effect? Osmosis, remember, is the diffusion of water towards a high solid concentration. Cells have macromolecules that are charged. They attract ions, counter ions from outside. This causes a high ion concentration in the cell. Water will come in, the cell will burst. But there is a solution. Three solutions are possible. What do plants, fungi, and bacteria do? I already told you, plants have a cell wall. Fungi have that too, bacteria have that also. The cell wall gives counter pressure. So when the cell takes on water by osmosis, the counter pressure from the cell wall simply stops that osmosis. That's just a new equilibrium that is reached. And because of that, we have turgid plants. So the rigid cell wall puts pressure that counteracts the effect of osmosis. So osmosis stops at some point. Now we still need proteins in the cell membranes of plants, fungi, and bacteria in order to make sure that the ions that come in and that stay in are the right ones. So we still have specific proteins. What is our solution? Our solution is our friends, the sodium-potassium pump. The sodium-potassium pump ensures that the excess ions that would get into our cells because of the Dolan effect are pumped out. But you're telling me not only sodium is pumped out. So what happens with the rest? Well, the sodium ion gradient is the driving gradient for the export of other ions in secondary active transport processes. So we use the sodium-potassium pump and the energy stored in ATP. 1/3 of all the ATP that we make goes to this purpose. It cannot be ignored. So we make a sodium gradient, and then additional proteins by secondary active transport are there in the membrane to regulate the exact ion composition, solute composition that we have. There's a third solution that some microorganisms use. Some microorganisms, protozoa, have a vacuole that's a contractile vacuole. It's actually a place where ions are sequestered, water follows, and this vacuole is then emptied by mechanical force. So the contractile vacuole also removes excess water and ions. We still need specific proteins to regulate exactly the ion composition in the cytosol. So I think you should know the 3 solutions to a problem caused by the Dolan effect. Let's never forget it. Okay, so the next time you think about the sodium-potassium pump, you are very thankful that it's there. Because you wouldn't exist without it. Okay, so the pumps set up, they serve, the sodium-potassium pump in our case serves to deal with the problem caused by the Dolan effect. But it's doing a little bit more. It's setting up a driving gradient that allows secondary active transport to work. So in our case, the sodium-potassium pump is causing that driving gradient. And remember, sodium is always outside, exoplasmic. So there we have our animal cells, simple diagram. Relatively low sodium concentration inside, millimolar amounts, a couple of millimolar. On the outside, we have over 100 millimolar sodium. So that's between a 10 and a couple of 100-fold concentration difference. So what if you don't have a sodium-potassium pump? Plants don't have that. Most fungi don't have that. If you don't have that, then what do you use to drive transport of other molecules? In that case, you use our friend the proton, so the hydrogen ion. So virtually all other systems, and all is true for eukaryotes, all other systems use proton gradients. So think about plants and fungi, and most bacteria that we study, but not all, they have a proton pumping system at their plasma membrane. And it's also a P-type pump. But this time it's pumping just protons. So this sets up a gradient that is used for the uptake of other molecules. So the P-class pump in the plant and fungal membrane. But that's not the end of it. Internal membranes also have ion gradients. And always the proton gradient is the most important for internal membranes. That's true in every biological system that I know of. And in the case of mitochondria and chloroplast phytoids, the driving gradient is formed by an electron transport chain, which is in the mitochondrion driven by the conversion of larger molecules to smaller molecules, and in the thylakoid membrane driven by light. So that's actually the mechanism of respiration and photosynthesis involves the formation of a proton gradient. Importantly, for these systems, we'll later see that that proton gradient is used to actually synthesize ATP. The bacterial inner membrane also has electron transport chains that generate these proton gradients. So I think you should know animal cells, sodium gradient, very important. More sodium out and that's it. Every other membrane system, it's a proton gradient, where the pH is always lower on the outside, the exoplasmic side. Proton concentration is higher, the pH is lower on the outside. And we can take that and apply it to internal membranes, where the exoplasmic, the non-cytoplasmic side of the membrane is always more acidic. Very important that you remember this. I think this is as important as knowing that DNA encodes for proteins. Okay, so think about our plasma membrane. We have a sodium gradient and a potassium gradient. What's causing that? The primary cause is this sodium-potassium bond, depicted here. Very simple diagram. ATP is hydrolyzed, free sodium move out, free potassium move in. Sodium-potassium ATPase, sodium-potassium bond. So this gradient of sodium and of potassium now allows for other things to be transported, such as maybe glucose that can come in from a low concentration in your intestinal gut lumen up to a high concentration in the intestinal cell itself, using 2 sodium. Okay, so we call that secondary active transport. And it allows the transport of other molecules against a concentration gradient. You can ask the question, why is this uptake of glucose not directly coupled to ATP? And I want to answer that question on Monday. For now, I'll tell you that this system is just more efficient. All right. I hope you'll have a good Valentine's Day.